We shall start the session with a simple question. What is a cell? A cell is a basic structural and functional unit of life. We have studied prokaryotic and eukaryotic cells. To restate, prokaryotes are primitive organisms or single-celled, whereas eukaryotes are organisms containing membrane-bound nuclei along with organelles that are otherwise lacking in prokaryotes. Interesting, isn't it? Let's study the structure and organization of eukaryotic cells in detail now. Eukaryotic cells are found in protists, fungi, plants, and animals. These are highly complicated and contain extensive compartmentalization called cell organelles. Imagine the eukaryotic cell as a house having different rooms like the kitchen, bedroom, living room and bathroom, and each room has its own function. Similarly, a eukaryotic cell has different parts within each organelle performing different functions, and these are enclosed by membranes. Eukaryotes have a true nucleus. So, what does this infer? The genetic material or chromosome is embedded within a membrane-bound nucleus. Eukaryotic cells in plants are different from those of animals as they possess a rigid cell wall, placids, and a large vacuole which is absent in animal cells. And animal cells have centrioles that are absent in plant cells. In this video, let us study the cell membrane and the various cell organelles. The endomembrane system comprises the various membrane-bound organelles which work together to perform synchronized functions such as packaging and transport of various biomolecules. They are present inside the cell. These include vacuoles, the nuclear membrane, lysosomes, Golgi complex, vesicles, and the endoplasmic reticulum. Although the plasma membrane is not located inside the cell, but is also included as an endomembrane system because it acts as a gateway for the packaged biomolecules out of the cell. It should be noted that the mitochondria, chloroplasts, and peroxisomes do not coordinate with the above organelles, hence they are not included in the endomembrane system. So, let us learn about cell membrane first. Can you recall the other name for cell membrane? Yes, it is also called plasma membrane. The structure was extensively studied on RBCs by using some chemical methods. So, what constitutes a cell membrane? Lipids and proteins constitute the cell membrane. The lipids are arranged in a bilayer consisting mainly of phospholipids and some cholesterol in such a way that the polar heads are directed outwards and the hydrophobic tails are directed inwards to protect them from the aqueous environment. In addition to lipids, it was also found that the cell membrane also contains proteins and carbohydrates. Interestingly, the ratio of protein and lipid varies in different cells. The membrane proteins are of two types: integral proteins, which are present partially or completely covered inside the membrane, and peripheral proteins, which are seen on the surface of the membrane. The fluid mosaic model of the cell membrane is a widely accepted improved model proposed by Singer and Nicholson. The model proposes the cell membrane to be semi-fluid since it allows the movement of proteins within it. Why is the fluid nature of the cell membrane given so much importance? Have you ever thought of it? The functions such as cell growth, endocytosis, secretions, formation of intercellular junctions, and cell divisions are regulated by this fluid matrix. So, what is the important function of the cell membrane then? It is to transport the molecules across it. The membrane is selectively permeable in nature and allows only certain molecules to pass through it. So, for the movement of particles across the cell membrane, various types of movements are involved. We shall discuss them now. Simple diffusion is a type of movement of molecules along the concentration gradient. Osmosis is a movement of solvent through a semi-permeable membrane. Then passive transport is the movement of particles without the need for energy. And now, active transport is the movement against the concentration gradient of molecules with the help of energy. Fungi and plant cells have cell walls, which are rigid and non-living. It is the outer covering. When we look into the structure of cell wall, it consists of primary cell wall found in young plants and is capable for growth, which diminishes as it matures. Secondary cell wall is present on the inner side of the cell. The outer side is composed of middle lamella made of calcium pectate, and this glues the neighboring cells together. The cell wall is made up of cellulose, hemicellulose, pectin and proteins, while in algae, the cell wall is made up of cellulose, galactans, mannans and calcium carbonate. Now let us discuss the functions of cell wall. Cell wall adds shape and structure to the cell. It also protects the cell from mechanical damage and infection. It facilitates cell-to-cell interaction and it also acts as a mechanical barrier. We have just discussed the plasma membrane being selectively permeable. Can you tell me about the permeability of the cell wall then? Well, the cell wall is permeable. Sometimes the plasmodesmata may traverse the cell wall and the plasma membrane connecting the cytoplasm of the neighboring cells. Now, let us talk about something interesting. There are some coiled structures inside the cell. They are called endoplasmic reticulum. These are tubular networks in the cytoplasm. The endoplasmic reticulum divides the cell into two compartments. They are the luminal part and the extraluminal part. That is the cytoplasm. If you check the image on the screen, you can see ribosomes attached to the endoplasmic reticulum. Such structures are called rough endoplasmic reticulum or RER. On the other hand, if ribosomes are absent and appear smooth, they are called smooth endoplasmic reticulum or SER. Let us now check the functions of endoplasmic reticulum. Rough endoplasmic reticulum is involved in protein synthesis and secretion. They are continuous with the outer cell membrane. Smooth endoplasmic reticulum is involved in the lipid synthesis. Example, in animal cells, lipid like steroidal hormones are synthesized in the smooth endoplasmic reticulum. Endoplasmic reticulum also provides the structural support and frameworks to the cell. They help in the transport of proteins and fats. Have you seen a flattened disc-shaped structure? They are the Golgi apparatus. This structure was first observed by Camillo Golgi in 1898 and thus named after him. In plants, they are called dictyosomes, which are small units. It is a structure that consists of many flat disc-shaped sacs called cisternae, stacked parallel to each other, arranged concentrically around the nucleus, with the forming phase or the convex cis and the maturing phase or the concave trans. And these two phases are interconnected. The movement of materials inside the cell and between the cells is facilitated by the Golgi apparatus. Thus, the Golgi apparatus helps in the packaging of materials to be transported either intracellular or intercellular or out of the cell. Golgi bodies remain in close association with the endoplasmic reticulum, since the packaged materials travel in vesicle via the endoplasmic reticulum to the cis phase of the Golgi and out of the trans phase. Another function is that the proteins synthesized by the ref endoplasmic reticulum are modified in the cisternae of Golgi body. Golgi apparatus is an important site for the formation of glycoproteins and glycolipids. Let us now check another cell structure, that is lysosomes. Lysosomes are membrane-bound vesicles formed by the process of packaging in the Golgi bodies. The isolated lysosomes are rich in almost all types of hydrolytic enzymes like lipases, proteases and carbohydrases, which are active at acidic pH. Let us now check the functions of lysosomes. Lysosomes help in the intracellular digestion. They destroy the foreign substances which harm the cell. They remove dead and worn out cells, hence they are also called suicide bags. They have the property of digesting stored food when food is unavailable to the cell. Have you ever noticed a big bag-like structure in the plant cells? They are named vacuoles. Vacuoles are membrane-bound clear spaces in the cytoplasm. It has a single membrane called tonoplast. The vacuoles in plant cells are quite large compared to that of animal cells and are filled with a liquid inside called the cell sap and other waste particles. The tonoplast facilitates the movement of materials against the concentration gradient into the vacuole. Now, let us point out the functions of vacuoles. They store water and other substances such as waste products and unwanted particles. Vacuoles provide turgidity to the plant cells by pressing against the cell wall. And vacuoles also contain pigments like anthocyanins. Have you heard of contractile vacuole in amoeba? It helps in osmoregulation and excretion of waste products from the amoeba. These are but a few structures and functions inside the cell. There are many more organelles performing different functions such as mitochondria, plastids and the nucleus. Watch on to learn more about each and how they coordinate, which makes a cell the unit of life. Now, let's summarize what we have learned.
Manorama Horizon. Learning made simple.